Folks, again, just for the movie, this is Thursday, November 14th, and we're doing 2 activities in lab. We're doing our follow-up on our Chapter 14 Kirby-Bauer antibiotic sensitivity tests, and then we're going to be inoculating our Chapter 16 metabolic tests, right? So we'll do 2 separate movies here. So folks, first of all, just kind of like essentials, it's like, what do we need to know for the lab exam, which I totally understand. So first of all, folks, we want to identify the targets of the antibiotics we used. And folks, this actually is going to be really helpful because you're going to see antibiotics on the comprehensive lecture final exam. So these last few labs, I think, are actually going to help in getting you ready for the lecture final. Okay, folks, so to which big class of antibiotics does penicillin and ampicillin belong? Beta-lactam antibiotics, so that's important to know, folks. What is the target of beta-lactam antibiotics? How do they inhibit or kill bacteria? Oh, I like that, right? What a great description. Irreversible, competitive inhibitors of bacterial transpeptidase. Okay, so folks, we know the target. What's the consequence? If bacterial transpeptidase can't function and the bacterium is growing in a hypoosmotic environment, what's going to happen to the bacterium? Lysis, osmotic lysis. Why? What's the function of bacterial transpeptidase? Cross-linking what? Cross-linking peptidoglycan. And where is that in a bacterium? In the cell wall. Who cares if the peptidoglycan is not cross-linked? What happens to the strength of the peptidoglycan if it's not cross-linked? It's weakened, right? And thus, it can't prevent osmotic lysis if the bacterium is growing in a hypoosmotic environment. Good, you guys. That was kind of a spiel, also kind of to get you ready for the final lecture exam. Good job, you guys. Now we have 2 beta-lactams, penicillin and ampicillin. What's the significant difference between penicillin and ampicillin? Good. If I use the term narrow spectrum and broad or extended spectrum, how would you use those terms to describe penicillin? Narrow spectrum, right? It's going to target primarily gram-positive bacteria. Why can't we use it against most gram-negative bacteria? The outer membrane. What about the outer membrane? Yeah, the outer membrane, lipid bilayer diffusion barrier, right? So what must water-soluble antibiotics pass through to pass through the outer membrane? The porins, the outer membrane porins, that's right. So you guys, can penicillin cross through the outer membrane porins of most gram-negatives? Most of them no, right? So that's why we're going to use them primarily for gram-positives. Is the gram-positive cell wall hydrophilic or hydrophobic? Hydrophilic. Remember, thick layers of peptidoglycan, water-soluble chemicals just diffuse right through it. So you guys, just while we're going here, you can see below our table I have mechanisms of resistance. And this is something we also need to know for the lab exam and for actually lecture exam 3. So you guys, I'll try to kind of list some mechanisms of resistance as we go. So you guys, so one mechanism of resistance would be that the cell wall acts as a diffusion barrier against the antibiotic. So what did you just tell me? Which of the bacteria did we just discuss has the outer membrane and prevents diffusion of some antibiotic? The gram-negatives, right? But you guys, can you think of another bacterium whose cell wall is the most protective against water-soluble antibiotics? Acid-fast, right? Remember, you guys, the acid-fast bacteria, like Mycobacterium, have that hydrophobic, waxy, mycolic acid-rich cell wall. You know, really, really high-level protection. Good job, you guys. All right. And then with ampicillin, what's different about ampicillin compared to penicillin? It's broad spectrum, and what does that mean? Broad spectrum, extended spectrum, both gram-positives and gram-negatives, and why? What's different about ampicillin? It can cross through the outer membrane porins, right? They chemically modified it, so now it can cross through the outer membrane porins of most gram-negatives. Good, you guys. So ampicillin, broad spectrum, extended spectrum, both gram-negatives and gram-positives. Good job, folks. What about tetracycline? What's the target? 70S ribosomes, right? The bacterial cytoplasmic ribosomes. What function will be stopped if a bacterium is sensitive to tetracycline? Translation, good. What's a simpler description of translation? Protein synthesis. Good job, you guys. And folks, just out of, I mean, to me it's kind of interesting. Tetracycline was an antibiotic commonly added to animal feed. Animal feeds as a growth promoter, yeah, because it's relatively broad spectrum. Okay, and then folks, erythromycin is a macrolide. What's the target of macrolides? 70S ribosomes. Good. Clindamycin, folks, is a demo. I don't think we described it in lecture. It's similar to the macrolide, similar to erythromycin, right? So what would be the target? 70S ribosomes, yeah. The reason I threw this in here, you guys, is that giving patients clindamycin might increase their risk of developing Clostridium difficile, pseudomembranous colitis. So some of the antibiotics, it seems like it puts your patients at higher risk for developing those C. diff infections, right? So I just wanted to throw that in there because I thought that was an interesting tidbit. So folks, we're adding to our table. It's not on the table. We want to add it. We're going to add gentamicin as a demo, and it's because it represents another class of antibiotics, the aminoglycosides. So folks, what are the targets of aminoglycosides? 70S ribosomes. So folks, remember how we talk about having default answers on our lab exams? It's like if we're so tired and we have no clue, you know, and I ask you, what's the target of this antibiotic? What would be a pretty decent guess? 70S ribosomes, right? Because I think most of you are really comfortable with beta-lactams going for the transpeptidase. So that, you know, I would use that as my default answer. It's like, I have no clue. I'm going to put 70S ribosome. I think that's a good guess. Good. And folks, one thing with gentamicin and all the aminoglycosides, one thing you have to be careful about is they can be really hard on your patient's kidneys. They can actually cause a little bit of kidney damage. So again, you'd want to be really careful if your patient is suffering from what underlying disorder. Yeah, any kind of kidney disease, be really careful giving the immunoglycosides. We'll presume you guys, our patients are all young and healthy and have no underlying kidney disease. Good. And then folks, the SXT is the combination of trimethoprim sulfamethoxazole. What's the target of trimethoprim sulfamethoxazole? Good. Sequential inhibitors of the bacterial enzymes involved in what? Folic acid synthesis. And you guys, folic acid is required for DNA and RNA synthesis, okay? So you guys, why is it that when we're given trimethoprim sulfamethoxazole that we don't suffer from a lack of folic acid? What's the difference between bacteria and humans? We don't have the enzymes to make folic acid, so we have to ingest it as a vitamin, right? But you guys, this is so interesting. Nina in the other section was saying that like pregnant women are warned, or not warned, you have to be careful giving like trimethoprim sulfamethoxazole to pregnant women because it might decrease their folic acid levels, and that could lead to, you guys in A&P, help me out, is it spina bifida? Yeah, okay. And I was thinking about, I go, now wait a minute, why should that be? This is my hypothesis, you guys. Remember the good bacteria growing in our intestinal tract? Some of them are making vitamins for us, folic acid, right? So if you're killing all the bacteria, making folic acid for you, right, that's going to deplete your supply of folic acid. So again, I thought, wow, that's fascinating. We don't think about that the bacteria in our intestine are helping us, for example, making vitamins for us. So I thought that was fascinating. Okay, good, you guys. So we've gone through the mechanisms. Would those mechanisms be fair game to ask you on the lab exam? Yes. Okay, good. And then, folks, I thought what we would do before we go through our data is look at some of the mechanisms of resistance, because you can see up there many of our bacteria are resistant, and we want to understand why. How is it that they can grow in the presence of these antibiotics? Okay, and on page 3 of your Kirby Bauer handout, folks, I've listed some of the mechanisms of resistance, and I think I might have like one or two more to add. So, folks, we've already talked about how the cell walls of the bacteria might act as diffusion barriers. So we said the gram-negative bacteria with the hydrophobic outer membrane, that's protective, right? And we said the acid-fast cell wall is really protective. Good. Okay. Then another mechanism, folks, is the bacteria might make enzymes that can hydrolyze, destroy the antibiotic. Okay, so let's do that. I think I'm splitting the enzymes up into two categories. So, hydrolytic enzymes. Destroy the antibiotic. Can you think of one we've discussed in lecture? You guys, remember, all Staphylococcus aureus, just presume they're all making what? Which enzyme? Remember Alexander Fleming said after he discovered penicillin, he said, be careful, if you overuse this, you're going to select for, penicillin-resistant bacteria, and they were really worried about Staph aureus. So you guys, do you remember the enzyme that probably all clinical isolates of Staphylococcus aureus make that makes them resistant to penicillin, ampicillin, amoxicillin? You remember the name of the enzyme, you guys? That destroy beta-lactam antibiotics? Beta-lactamase is excellent. I mean, not excellent for us, but you are right on. Okay. And then, folks, a mechanism we might have just mentioned in lecture is that some bacteria will make enzymes that don't destroy the antibiotic, but enzymes that will chemically modify the antibiotic. For example, adding a functional group. So let's put enzymes which add functional groups to the antibiotic, and then the antibiotics can no longer bind to their target. And I think a relatively common bacterial enzyme are methylases. These are bacterial enzymes that would add a methyl group, and a methyl group, you guys, it's carbon with 3 hydrogens. Okay, and then when the antibiotic is methylated, it can't bind to its target anymore, right? And then, folks, let me see here. Then we can have a mutation of the target. And folks, for this one, let me give you the example of MRSA. What does MRSA stand for? Methicillin-resistant Staph aureus. And you're like, now remember, methicillin is supposed to be resistant to beta-lactamase. So how is it that MRSA can grow in the presence of methicillin and all beta-lactams? Excellent, you guys. Excellent. A mutant transpeptidase, right? The target of the beta-lactams. MRSA have a mutant transpeptidase to which none of the beta-lactams can bind. All right? And then, folks, another really disturbing mechanism are antibiotic efflux pumps. What is an antibiotic efflux pump? Yeah, they're protein pumps in the cytoplasmic membrane that do what? Pump out the antibiotics. And remember, you guys, sometimes they're non-specific, meaning they could pump out more than one antibiotic, yeah? And folks, I just, it's in your handout, but I thought another one that we could add here. Can you think of a really, really resistant structure that some bacteria make, like the genus Bacillus and the genus Clostridium, that are totally resistant to antibiotics? Endospores, awesome, good job. And remember, you guys, our endospores, we're going to find them made by pathogenic strains of Bacillus, like Bacillus anthracis, the anthrax endospores, and also by members of the genus Clostridium. And there's many different Clostridium, you guys, and they all make endospores. So Clostridium difficile, right, that causes diarrhea, pseudomembranous colitis. Clostridia tetani causes what? Tetanus. Clostridium botulinum causes botulism, good. Clostridium perfringens, you guys, can cause gas gangrene, like when people have injuries that aren't cleaned, they can develop a gangrene, right? And often that can be caused by members of the genus Clostridium. Good. So you guys, would antibiotics destroy an endospore? Nope. They're going to laugh at you. Would an alcohol hand sanitizer destroy an endospore? Nope. They're going to laugh at you, right? So we just want to remember those endospores. Those are tough. Good. All right, you guys. So those are kind of some basics for both the lecture and for lab. So now let's look at our data, okay? So you guys, it will be important for the lab exam that you know the bacteria that we tested, are they gram-negative or gram-positive, right? Because that's going to influence their sensitivity or resistance. So you guys, E. coli that you tested, what kind of cell wall does it have? Gram-negative, right? So you would predict of the antibiotics that you tested, even before you got your data, you would predict it was going to be resistant to which antibiotic? Penicillin, right? So were you right? Uh-huh. So you guys, we would suggest that the E. coli is naturally resistant to penicillin because of the outer membrane of the cell wall, right? Diffusion barrier. Good. But now this is where it gets interesting, folks. So before you ran the experiment, what would be your prediction about E. coli sensitivity to ampicillin? Would you have predicted? Yeah, I would have predicted ampicillin could cross the outer membrane, right? But you guys, what did we get? Yeah, and folks, in a weird sort of way, this is really cool because bacteria can have more than one mechanism of resistance. My guess is our E. coli is making a beta-lactamase. A lot of gram-negatives make beta-lactamases. I'm guessing that our E. coli is making a beta-lactamase, which is really disturbing because we basically don't want you guys working with bacteria that are actually carrying antibiotic resistance genes, right? So again, you guys, make sure you're washing your hands really good before you go. All right, good. And then, folks, tetracycline, we have this intermediate resistance. And again, gosh, this makes it really challenging because you'd have to look at the whole clinical picture. Like, is the patient that's infected, are they strong? You know, immunocompetent, strong patients, no underlying diseases. And the second thing you'd want to look at, folks, is there another antibiotic available to which your bacteria is truly, truly sensitive, right? If you're in a situation where the only antibiotic that shows any kind of ability to inhibit the bacterium is one that has intermediate, I guess you'd go for that. But you guys, this would be horrible if, say, your patient was really immunocompromised. Say it was an HIV/AIDS patient. The patient's immune system can't contribute anything to try to kill the bacterial pathogen, right? So this one, I would sure be looking for another antibiotic that can outright kill, right? So you guys, so we got that on erythromycin. You guys, I think the kind of the average of the data, it's also intermediate. So let's just put it intermediate here, right? And what about the clindamycin? Resistant, right? Oh my, this is not good, you guys. This E. coli, holy smokes. Oh, whew. What about gentamicin? Is it, okay, this was the demo data, you guys. This E. coli is sensitive to the gentamicin, so this would be a possibility if the patient doesn't have an underlying kidney problem. Good. And folks, with the trimethoprim sulfa, we have a mixture of intermediate and sensitive. So this is my thought, you guys. I would probably put the gentamicin is the first choice as long as the patient has no underlying kidney issues. And then if they do, maybe consider the trimethoprim sulfa. Does that make sense? Okay, so maybe we could rank them, you guys. Maybe we'd go first, the gentamicin, and second, the trimethoprim sulfa. Are we okay with that logic? Yeah, I think for a lot of you it's just intuitive. You just want to make sure everybody feels comfortable with this. Good. You guys, the Pseudomonas, the reason we wanted to give you Pseudomonas fluorescens is there is a cousin of Pseudomonas fluorescens called Pseudomonas Aeruginosa, which is a really, really sometimes devastating opportunistic pathogen. It can cause really serious infection in cystic fibrosis patients. It can form biofilms in the lung. And we know biofilms make it even harder to treat, right, if the bacteria are living in biofilms. And we're going to see it's just naturally pretty resistant. It's really a bad news, you guys, in your burn victims, right? And so we didn't want you working with Pseudomonas aeruginosa, so we gave you a less virulent cousin, Pseudomonas fluorescens. And again, you guys, the reason is we wanted you to see how resistant Pseudomonas can be. And again, they're gram-negative, so they have an outer membrane. And probably the Pseudomonas outer membrane porins are really good at excluding antibiotics. So it's really hard to get the antibiotics across the Pseudomonas cell wall. So you guys, so let's take a look at our data. So with penicillin, it makes sense that it would be resistant, right? It's a gram-negative. And the ampicillin, let's see here. Let's see. Did you guys run Pseudomonas? Did you actually run the Pseudomonas? Oh, so this is just the demo data? Okay, got it. Understood, you guys. All right, I was just like, wait, did we run Pseudomonas? Okay, so you guys, in ampicillin, right, again, this is I'm thinking this is a reflection that in Pseudomonas, again, the outer membrane porins exclude so many antibiotics. So this might be a reflection simply that even the ampicillin can't cross the outer membrane. So we're going to go for resistance here. Thank heavens, folks, that the tetracycline that targets the Pseudomonas 70S ribosomes, it's crossing at hopefully therapeutic levels. So we could use tetracycline, right? So that's good news. And the erythromycin, either the erythromycin can't pass through the outer membrane, or maybe the Pseudomonas has some enzyme that can modify the erythromycin so it can't target the ribosome, so resistance. Clindamycin, the same thing, resistance. Again, folks, luckily, the gentamicin, okay, so far we have two, you guys, the tetracycline and gentamicin, and resistant to trimethoprim sulfa. So folks, if this was, say, a cystic fibrosis patient that has a serious Pseudomonas infection, which antibiotic or antibiotics could you suggest? Yeah, the gentamicin. So gentamicin, so our data supports. So what would be the two antibiotics, you guys? Good, the gentamicin and tetracycline. I'll put tetracycline because I think it's a little less hard on kidneys. But folks, do remember if this is a cystic fibrosis patient, which, I should add that, you guys. I should add that to resistance. What's another way that bacteria can resist antibiotics? Growing in biofilms, right? And remember, you guys, Pseudomonas and cystic fibrosis patients in the lungs, they're very often growing in biofilms. And we know that the layers of biofilms make a diffusion barrier. Often the bacteria in the lower layers are in the stationary phase of growth. The layers of the biofilm are a diffusion gradient against antibodies, immunoglobulins, and also the phagocytic cells can attach. So let's add that, you guys, growing in biofilms. Good. And then, folks, your Staphylococcus epidermidis, this is a stand-in for which serious gram-positive pathogen? Staph aureus, right? And Staph aureus, you guys, is very aggressive. And we'll discover in the medical microsection of lecture, it has so many virulence factors. It can spread so rapidly throughout our body. Very aggressive. Can be a hospital-acquired pathogen, which is bad news because often it's resistant to multiple antibiotics. So you guys, let's look at our staph epi. So would you have predicted, without running the experiment, that Staphylococcus epidermidis might be sensitive to penicillin? Without running the data. So you guys, so you're thinking, okay, wait a minute. So Staphylococcus epidermidis, is it gram-negative or gram-positive? It's gram-positive. So should its cell wall prevent any protection? No. So you guys, before we ran the experiment, we would have predicted, just based on the cell wall, we would have predicted it should have been sensitive, right? But you guys, we're going to go with, it's kind of split here. So you guys, okay, so some of our results say it's sensitive, some say resistant. You guys, let's just explore what if it was resistant. And this is the data the Monday, Wednesday class got to, is the staph epi was resistant to penicillin. So you guys, if it's not the cell wall, what do you think is the most likely explanation? Exactly, beta-lactamase, right? Does that make sense, folks? Yeah. And folks, if the staph epi has a beta-lactamase and is resistant to penicillin, what's your prediction about ampicillin? The staph epi should be likewise resistant to ampicillin, right? Because beta-lactamases destroy penicillin, ampicillin, amoxicillin. Good. And did we get that? We got kind of the same mixed results, you guys, but let's, just to make this more dramatic, let's go for resistance, okay? And again, this would be consistent. If we have a beta-lactamase, it's going to destroy penicillin and ampicillin. Good job, folks. What about tetracycline? Yeah, that's good, you guys. So at least we have tetracycline. What about erythromycin? Nope, not going to use that one. What about clindamycin? Nope. Right? What about gentamicin? Yeah, that's definitely a possibility, you guys. Let's go sensitive. Good. And what about our trimethoprim sulfa? Resistant, yeah. So you guys, if our data for our staph epi, which antibiotic or antibiotics would you suggest? Yeah, so we're going to go tetracycline. And what's another one, folks? The gentamicin. Wow, okay. Wow, you guys, that's great, because if I ask you on, wait, we should have done up here. Wait, I should have done E. coli. Okay, sorry. Let me do the E. coli. So with E. coli, you guys, what were the two that we said? The gentamicin. That was intermediate. It's like the best was the gentamicin, right?
Yes.
Okay.
And then your second one was the Bactrim.
The Bactrim was, yeah, kind of, yeah. We'll put it in parentheses because it was intermediate there. Okay, so you guys, in our class data, which antibiotic was effective at therapeutic levels to kill our gram-negative E. coli, our gram-negative Pseudomonas, and our gram-positive Staph aureus? The gentamicin, right? And often it is described as a big gun, but remember, you guys have to be careful because you can cause kidney damage and auditory nerve damage too, okay? Awesome, folks. With our Pseudomonas and our Staphylococcus aureus, you guys, which two antibiotics at therapeutic levels would inhibit the Pseudomonas and the Staphylococcus? The tetracycline and gentamicin. Good. So, folks, looking at this data, can you tell me, is gentamicin narrow spectrum or broad spectrum? Good. Excellent. And, folks, what about tetracycline, the antibiotic that's often added to animal feeds? Is it narrow spectrum or broad spectrum? Broad spectrum, right? Good job, you guys. That's beautiful data. Okay, so just to finish, you guys, let's look at our Lab Exam 2 study guide packet and finish the Chapter 14 practice exam questions. And this is going to start, folks, on page... let's see here, 9... Yeah, on page 10, folks, I think, up at the top where it has the matching. Does that make sense? The matching between the antiseptic disinfectants. I think we gotten through that part, right? Okay, so you guys, let's start with question 15. Which structure, if any, protected Staphylococcus epidermidis from water-soluble antibiotics? D, none, right? There's no protective structure in the cell wall. Good. 16. Which structure, if any, protected E. coli and Enterobacter aureogenes from water-soluble harmful chemicals? A. Good. The outer membrane. Good job, folks. 17. Which unusual protective structure can Bacillus subtilis make, which Staphylococcus epidermidis cannot make? What a mouthful. C. Endospores. Good. 18. Clostridium difficile, which causes pseudomembranous colitis, makes this protective structure, which is not inactivated by alcohol hand sanitizers, and we can add, you guys, nor by antibiotics. What would that be? C. Endospores. Good. Number 19, folks. Which structure, if any, can protect Mycobacterium tuberculosis from water-soluble antibiotics and other harmful water-soluble chemicals? E. The acid-fast cell wall. Remember, you guys, that's like of cell walls, of bacterial cell walls, the acid-fast cell wall is the most protective. Good. So, folks, 20, which mechanism below is not a strategy used by bacteria for antibiotic resistance? D, yeah. It would not help a bacterium if it had a mutation which increased diffusion of the antibiotic across the cell wall. So, you guys, so for question 20, D is the correct answer. So, folks, 21, which of the following may be used by bacteria to horizontally spread antibiotic resistance genes? D, all of them. So, specialized and generalized transduction, transformation, conjugation, all of those. And again, you guys, like questions 20 and 21, those might even appear on lecture exam 3 because we actually did talk about horizontal gene transfer, the transfer of antibiotic resistance genes, and we did talk about antibiotic resistance mechanisms. Good. So you guys, 22, match the action of the antibiotic, that is the mechanism of inhibition, to the antibiotic. So you guys, Trimethoprim Sulfamethoxazole, it would be sequential inhibitors of folic acid. Good. Penicillin, A, inhibits transpeptidase. Ampicillin, A, erythromycin. C, inhibitors of bacterial translation. So remember, you guys, you knock out the 70S ribosomes, you knock out translation protein synthesis. Tetracycline, C. Methicillin. It's actually A, because remember, you guys, methicillin is a beta-lactam, so it's going to target the bacterial transpeptidase. It's resistant to beta-lactamases. That's what made it so wonderful. And you guys, the last one, Carbapenems, if I ask this on the lab exam or the lecture exam, it would be a bonus question. So Carbapenems, you guys, they're related to the beta-lactams. What makes them awesome is they can cross the gram-negative outer membranes, and they're resistant, or at least they were resistant to almost all the beta-lactamases that gram-positives and gram-negatives made. But did we overuse them? Yeah, And then we selected for bacteria resistant to carbapenems because they make carbapenemases. What do you think a carbapenemase is? Yeah, exactly. Hydrolyzes the carbapenems. Yeah. So these are the nightmare bacteria that we don't have any, a lot of them, we don't have any antibiotics left to treat with. Yeah. Good job, folks. 23, folks, I would ask you about MRSA. So what are MRSA? The methicillin-resistant Staph aureus. Why are they resistant to methicillin? The mutant transpeptidase. Awesome. You guys, I won't ask you about VRE, the vancomycin-resistant enterococci. But again, as a bonus, I could ask you about the CRE, the carbapenem-resistant Enterobacteriaceae. What do they make that lets them live in the presence of carbapenems? The carbapenemase is good. That would be a bonus question, folks. And then the last page for chapter 14 is just a practice. Kirby Bauer, disc diffusion. So it's just so you can practice at home measuring the diameter zones of inhibition and then practice using your interpretive chart. Okay? Yeah, so just to pretend. Okay, so folks, what I'm going to do now is stop the movie, and because it's already so long. And then what we're going to do, folks, is discuss what we'll do with our plates. And then I'm not going to record today the instructions for inoculating the Chapter 16 metabolic test because it's in your handout, but I will record next week our results. We want to discuss the results, okay? So let me just stop the movie really quickly. And folks, what you will need for this next experiment, you're going to need full PPE, so gloves, goggles, lab coat, lab shoes.